Okay, all right, why don't we go ahead and get started? So I just wanted to welcome everyone online to the Informatics Tools for the Pangenome Applicant webinar. I just want to first take this opportunity to thank my colleagues at NHGRI. I also want to thank our NHGRI IT team for setting up the webinar and our colleagues from NCI and NIA. Next slide, please. Okay, so I'm just going to do some quick introductions for those who will be talking this afternoon. We have Gabby Villard, who will be advancing the slides for us. Thank you. And we have a number of program staff on the call today. Myself, Idan Gabdunk and Ian Nova, Jerry Lee from NCI, Michael Benani and Marilyn Miller from NIA, and we have Sarah Whelan from our review branch. So as we move into the applicant webinar, I'll start by just giving some background information about the Human Pangenome Reference Consortium. I'll then hand it over to Idan to take us through the informatics tools for the Pangenome NOFO. Ian will then go over the SBIR and STTR tools NOFO. I'll end with a few administrative items, and Sarah will briefly go over changes in review. Just a few housekeeping items. The webinar will be recorded and made available on the NHGRI website probably in about 2 to 3 weeks. Any links that are mentioned in the webinar will be pasted in the chat for everyone to see. The chat has been disabled. So if you have a question during the Q&A, you can raise your hand. I will call on you and our IT folks will unmute you. You also have the option to put your questions in the Q&A box. So please, that's another way. And I think that is about it. So why don't we go to the next slide? Great, thank you. So I briefly wanted to go over phase 1 of the Human Pangenome Reference Consortium, starting back in 2019 through 2024. The goal here was to sequence 350 genomes from 1,000 genome samples. Activities included creating a useful pangenome resource for representation, developing a better way to represent a multi-genome reference, developing methods for assembly, integrating LC at all stages of the research, and fostering international partnerships through the Human Pangenome Project, which is a GA4GH derival project. Next slide. A major milestone from phase 1 is the paper seen here in Nature. So we have the first draft of the Human Pangenome Reference from 47 phased diploid assemblies from a cohort of genetically diverse individuals. Next. Applications of the pangenome include removing biases inherent to the use of a single linear reference genome, creating a community resource to aid in the development of methods and analyses of pangenome based population genetics, improve representation of tandem repeats, and improving RNA sequencing mapping. Next slide. So now moving on from phase 1 to phase 2, this cartoon is a visual abstract of what was proposed in phase 1. The top line shows the flow from sampling through sequencing, assembly, through pangenome development to community engagement and outreach. The second line is phase 2, which is similar, but with some noted changes in green. 200 new samples will be collected from the BioMe Biobank and Atlas Biobank. High quality genome assemblies will be added. A substantial embedded LC is still included from the current level in phase 1. So for this next phase, we wanted to significantly expand the outreach elements, and in particular, development of computational tools to allow broad use of the Pangenome reference and facilitation of adoption by the community through direct engagement. And again, just wanted to emphasize utility of the resource, that we're not just adding additional genomes. Next slide. Okay, so now I'm going to turn it over to my colleague, Idan, to take us through the Informatics Tools NOFO. Idan?
Thank you, Jyoti. So the NHGRI consortium is planning periodical releases of the Pangenome reference, making the need for tools that would allow scientists to work with the new reference more pronounced. The motivation behind the NOFO for Informatics Tools reflects the need to support both current functionalities that were available for linear genome, such as alignment, annotation, and also new functionalities that will be enabled by the nonlinear structure of the new Pangenome reference. Development of these tools is necessary to enable new research opportunities, as well as to promote wide adoption of the new reference Pangenome. Next slide, please. The consortium structure includes the following components: administrative and coordinating center that is based in WashU, data generating center that is based in UCSC, embedded LC component that was mentioned by Jyoti, and informatics tool development groups. The four groups that were founded last year focus on improvement of variant calling, visualization tools, compatibility with large-scale datasets, and aggregation of epigenetic information. It's important to point out that the embedded LC functions across all the components, advising on issues related to population labeling, data sovereignty, and population genetics. I would like also to emphasize that the different components of the consortium work together as a team, and we would like to see the new informatics tools be fully integrated within the consortium. Next slide, please. The funding opportunity we are discussing today is aiming to assist with uptake and make Pangenome useful to the scientific community, and to support creation of new tools that would take advantage of new types of information that are enabled by the Pangenome reference, as well as further develop existing tools to support new data types and support new use cases. I also want to say that any tools created should be relevant to broad sectors of scientific community and are expected to be openly shared, facilitating dissemination. Next slide, please. Here is a handful of examples of tools that would be considered in scope for this funding opportunity. Those include visualization tools for functional genomic features and structural variation within the context of a pangenome, comparative genomic analysis tools, tools for improvement of fine mapping and polygenic risk scores, and tools for genomic variance annotation. Next slide. The NOFO is co-funded by NHGRI, NIA, and NCI, each institute having different focuses and priorities, allowing for a broader range of applications to be submitted. In this case, NCI is interested in tools that would incorporate pangenome resources for somatic cancer analysis, and NIA in tools that will incorporate pangenome resources for aging and Alzheimer's disease or Alzheimer's disease-related dementia research. With that, I would like to ask Ian to continue on the next slide.
Yes, thanks. So new this round, we also have two funding opportunities specifically for small business focused research on this same topic. It's very similar in scope to the UL1 call for applications, meaning we are looking for new tools, taking advantage of new information, as well as existing tools modified to enable important use cases. The two small business opportunities are almost identical in text to each other, with one key difference about eligibility and where the work takes place. STTR projects require a collaboration between a small business and a research institution, whereas SBIR projects can either be a collaboration or a small business working on their own. In the funding opportunity, you will find more detailed information about eligibility and required company structure to be considered a small business, as recognized by the Small Business Association. Both of these opportunities are for phase 1 projects only. And in a phase 1, you are expected to demonstrate feasibility, technical merit, and commercial potential of your informatics tool. Phase 1 is designed to stage the product or service for a hypothetical future phase 2, where budgets are allowed, in a phase 1, budgets are allowed up to $400K total costs and durations for up to 1 year. Next slide, please. The timeline for these NOFOs is identical to the UO1 call, with a single receipt date on March 3rd, 2025. However, there are a couple key differences. These projects are focused on early stage development of a commercial product or service. This is different than a research project. If you read through the review criteria to understand some of the differences, one key factor is that innovation significance can be focused around making a tool more user-friendly, interoperable, and commercially viable to a broader community. However, again, I will emphasize this is phase 1 work, so the product and service can be at a very early stage and no prior data is required. The other key difference between small business and UO1 awards is that the small business awardees will not have the same consortia obligations as the UO1 projects, but they will be encouraged to attend the annual meeting. Thank you. Next slide.
Great, thank you, Anne. So I will take us through some administrative items. So for the timeline, letters of intent are due February 3rd. This is not mandatory, but it is helpful. It allows IC staff to estimate the potential review workload and to plan the review. As Ian mentioned before, applications for this NOFO are due March 3rd as well. The review will be sometime in June or July of 2025, convened by NHGRI. It'll go to our council in September 2025, and the early start date would be December 2025. The budget for the participating components, we intend to commit $3 million in FY26 for up to 6 awards. For eligibility, foreign institutions or foreign components are eligible. And for the NIA and NCI applications, it would be helpful to indicate that in your application, but we also do a check with NIH's receipt and referral. Next slide. So the application types, so this is open to everyone, open to all. It's also open to resubmission applications. This is applicants who applied from round 1 are allowed to apply as well. And we're also allowing competitive revision. And this is only applicable to current awardees. So please follow up with your program officer for competitive revision. So now I will turn it over to Sarah Whelan to talk about changes to the review criteria.
Okay, so hello. I'm not sure how much people have heard about the upcoming changes to applications and review. There are a couple of links now in the chat. There are multiple changes to fellowships and training applications. The most important ones for this, though, are the changes to the review criteria for the research project grants. This does not apply to the SBIR and STTR funding opportunities, but it certainly will be in effect for the UO1. What has happened is that the same 5 criteria, the significance, innovation, approach, investigator, and environment, will be evaluated, but they are reorganized into 3 factors. Really, the questions to be asked are, is the research worth doing? And are the investigators doing this correctly, and are they able to do this? So the factor 1 is the importance of the research, and this is the significance and innovation criteria. This will be scored from 1 to 9, 1 being the best as usual. Factor 2 is rigor and feasibility. This is the approach, which will also get a score from 1 to 9. And those both have, for this funding opportunity, those both have NOFO specific criteria that you should certainly pay attention to. Factor 3 is now expertise and resources. This is investigators and environment. The most significant change here is that factor 3 does not get a numerical score. This is just a drop down saying adequate or gaps identified. This factor is considered when assigning an overall impact score. But the idea is to downplay the role of investigators and environment and simply assess, are they able to do this work, and are their resources sufficient to do this work? And so when you are replying to this funding opportunity, there are no changes required to the structure of the application. That it will be exactly as you've always done for this type of activity code. But just to know that the reviewers will be assessing things a little bit differently.
Next slide. Great. Thank you. So for those of you who are not familiar with a UO1 mechanism, this is a cooperative agreement, which means that there will be substantial programmatic involvement with the recipients. So basically what that means is that we're working jointly with the award recipients in a partnership role. So we're not assuming direction or prime responsibility or a dominant role in the activities. Just wanted to point out a few things in the terms and conditions of the cooperative agreement, to collaborate with the steering committee members, which includes the Corning Center, the Genome Center, and phase 1 of the informatics tool recipients, to attend the annual in-person consortium meeting, actively participate in various working group calls, and accept and implement policies that are approved by the steering committee. Next slide. Okay, just a few reminders here. Again, we encourage you to read everything, but wanted to point out in section 4 for the application submission information, please send your LOIs and questions to Idan, and his email is here. Pay attention to the research plan. And then in section 5 for the application review information, as Sarah mentioned about the scored review criteria. So I just wanted to make a quick note about the process. So applications are assigned to the appropriate IC. Applications will compete for available funds in response to the NOFO. So following initial review, recommended applications will receive a second level of review by our advisory council, and then followed by, we'll consider everything for funding based on the scientific and technical merit as determined by scientific review, availability of funds, and relevance of the proposed project to program priorities. In section 6, as I mentioned in the last slide, please read through the cooperative agreement terms and conditions. There's also additional information about dissemination and sharing of tools. And I think next slide. So thank you for listening in to the webinar. So this concludes the presentation portion. And I think we can probably take down the slides and we can move into our Q&A session. So I don't see any questions right now, but I want to just open it up to my colleagues online. Is there anything additional that you wanted to mention that I may have missed, or any further details? Okay, so it looks like we have some questions online. How many applications will be funded by each of NHGRI, NCI, and NIA? So as I mentioned before, we have up to 6 applications, depending on what comes in, 1 from NCI, 1 from NIA, and we just have to see availability of funds. Is data analysis adapting pangenome technology suitable for this NOFO? Idan, would you want to maybe answer this?
Sure. Can you maybe clarify what you mean by adapting pangenome technology? It will allow me to answer the question more precisely.
So can we have maybe Juan Ping, would you like to unmute? Okay.
Can you hear me?
Yes, we can hear
Yes.
you.
Okay, cool. So, my question, because either for the NCI cancer genome or for the NIA Alzheimer's genome study, most of the time we will do the analysis, the genome sequence for the disease, mechanism. So, doing this, I mean the data analysis. This one is slightly different from the tool developed. So I wonder, is this NOFO specifically focused on the tool developed and exclude the data analysis part?
So this funding opportunity is definitely about tools that could be used for data analysis. Now, I could imagine I would let NCI and NIA colleagues to answer this question more precisely, but I could imagine that some of the tools could be developed to handle specific disease, and then it would probably go into one of those institutes, but I don't want to speak for them.
My name is Jerry Lee. I'm a program director in NCI's Division of Cancer Biology. I can briefly comment on your question. As my colleagues mentioned, that this particular RFA is really encouraging development of novel methods and technologies, taking advantage of the pangenome resources. If you're interested solely on using existing tools to perform data analysis, that wouldn't make the application particularly strong. If you, in the process of developing and improving informatics methods and propose to use that methods to do some data analysis as a sort of validation of the methods you develop, that would be deemed appropriate. I don't know if that answers your question or not.
Yep, thank you.
Yeah, if I may just add to Jerry's answer. So I'm Michael Benani, Program Director of Genetics within the Division of Neuroscience at NIA. So we have exactly the same view as Jerry's for NCI at NIA. The idea here for NIA is to use data that have been funded by NIA, mostly for aging and Alzheimer's disease and related dementia genetics and genomics as use cases for the development of tools and informatics. So yes, as Jerry said, once the informatics have been developed by the applicants, we would like them to use our funded data sets as use cases to show the feasibility and the power of the new tools as compared to what already exists. Marilyn, I don't know if you have anything you would like to add.
No, I think that's generally what we had discussed with our planning group and with what the director had in mind. So I think that's where we want to land.
All right. Wang, does that answer your question with you?
Yeah, thank you. Thank you. Yeah.
All right. Thank you.
Okay, we had a question in the chat about indicating which institute we're applying to, and Sarah posted an answer to the question. Yes, you should indicate the institution that you're applying, so the focus of your proposal will be assessed, and the application may or may not actually be assigned to the requested institution. There was another question in the Q&A about scheduling meetings with your program officer before the submission deadline. Yes, please do that. It's very helpful for us. Idan, if you could just maybe put your email in the chat, if you haven't already, that would be great. And so we have another hand raised. Am I pronouncing Xiaoling Zhang? Thank you.
Oh yeah, I have a similar question. Can you hear me?
Yes, we can hear you.
So since this genome that you guys just mentioned encourage new methods, new tools. However, there are some situations there are novel genetics components we can apply or modify our existing data to this pangenome data sets. And is that also appropriate? Not targeting specific disease or mechanism, but still globally.
Yes, the further development of existing tools to be applicable on pangenome reference is also fitting this funding opportunity.
Okay, thank you.
So Shaolin, from the NI perspective, any informatics tools that would increase the adoption and the use of the pangenome as a human genome reference for NIA genomics type of studies would be very, very welcome, because as of now, most genomic analyses in aging and Alzheimer's disease and related dementia are still using HG38 as a human genome reference.
Yeah.
NIA has a major interest in increasing genetic diversity type of analyses, which obviously the pangenome would offer. And so that's really where we stand with this NOFO.
Thanks.
And then there's another question. Can the tool developed be tailored for a specific condition? The example here is with Alzheimer's.
Yeah, I think as Idan explained, as long as the informatics that has been developed can be applicable to other fields of research, I can't imagine that an analytical tool or an informatics tool has been developed that can be used specifically and only to Alzheimer's disease. So as long as it's applicable to other areas, I think it will be acceptable to all ICs here. As far as NI is concerned, we need the use case to be derived from genomics and genetics data sets that have been funded by NIA, which in our case are mostly the NIGATs and the Alzheimer's disease knowledge portal.
Thank you. So we have a question Do successful applicants require previous expertise in tool development for pangenomes, or general expertise in tool development suffice? Yeah, go ahead.
I can attempt to answer this
Yeah.
question. Well, I think that pangenome reference is a very broad category of products that are being developed by the HGRP. So in general, I don't think that you have to be an expert in pangenome tool development to apply and be like, it's not required to be an expert in tool development specifically for pangenomes to apply. Sorry for the long answer.
And Sarah, did you want to add?
Yeah, I would say, I mean, the reviewers are just asked to assess whether the applicant and their team has the expertise to do what they plan to do. So as long as you have the expertise to complete your proposal, that's sufficient.
Yeah, let me just add, I think generally speaking, in the long term, what the field wants to do is to increase the number of investigators who can use the pangenome as a tool. So it seems a natural course to be flexible on this if we can.
These are all great questions. Thank you. Anything else? John Farrell raised hand. John, we can't hear you.
One of the issues blocking the use of pangenome tools is the lack of workflows to scale up to tens of thousands of genomes. Would workflow tools that deal with some of that scalability be something that would address this request? Or there's a lot of tools, but how you use them, for example, adding QC of project level variants, which aren't typically included in the available tools, things of that nature, imputation panels from resulting data sets, not necessarily a tool, but a resource that can be used by other researchers in their work.
So I think that, well, I'm not sure that I followed fully your question. One part of your question was about addressing scalability issues, and I think that's an important thing that has to be handled, especially as we add hundreds of new genomes to be a human genomic reference pangenome. That would need to be solved. So it makes sense to me as a relevant topic. But the second part of your question, I'm not sure that I understood.
regarding things like imputation panels, for example. We have imputation panels that do SNPs, but there's no, these lack structural variants that have been detected with pangenomes and stuff. With a resource like a reference panel, developing something like that, address this. And that would enable the use of the benefits of structural variant calling or these in areas that short read sequencing could be developed to address this in GWAS data sets as well as whole genome sequencing.
Thank you for the clarification. So you're asking if development of tools that will impute structural variants or things that are more complicated than SNPs, you're asking about those types of tools?
Yeah.
Yes, so it seems to be relevant and yeah.
Okay.
If I can just jump in for a second, the software is really for supporting informatics tools and method, as long as the main task of the project, 90% of the budget is spending on computational informatics method development, that'll be fine. But if the intention of appropriating a significant chunk of the budget, over 10%, wet lab or experimental methods and resources, that wouldn't be deemed very responsive.
Any other thoughts on that? We have another question. Will a tool addressing multi-ancestry population variants from the pangenome be considered responsive to the NOFO? I can take that on. I think yes, that would be responsive. And as mentioned before, please contact program officers. It's helpful to look at your specific aims and research proposal.
Yeah, we have the contacts for the three NH institutes, scientific contacts, contact information provided in the funding announcement. So if you have any questions, please go ahead, email us, and we'll be happy to get back to you, answer your questions.
So there's another question: is embedded LC research required? I can try to answer this, and maybe I'll ask Adam to pitch in. So the LC research is already incorporated in the consortium-wide research activities, but specifically for the tools NOFO. Adam, did you want to? I don't think it's required.
Yeah, Jyoti, it's not required. I can certainly imagine some tools that would be more or less directly related to LC, because LC is very, very broad and includes privacy preservation, things like that. That would be a reasonable thought, but no, it's not required. The LC embed is funded through the production center, and Jyoti's exactly right. It's already there.
Great questions. Thank you. Again, any questions, but maybe Sarah, or was there anything else that I know wanted to include, or anything additional you want to say about review?
Just the main thing with review is pay attention to the NOFO specific questions in section 5 of the funding opportunity, and pay attention to section 4 of the funding opportunity. Remember that that's how the reviewers will be asked to evaluate you.
Can you maybe mention for the applicants who are submitting an A1, how will their application be any different, since it was being reviewed under the older version
Right.
review criteria and with the new criteria?
Right. So the simplified review framework will apply to all applications in this meeting, even if it was, even if it is an A1 and was previously reviewed under different criteria. So they will all be reviewed with the same 3 factors. And there should be no real changes required to the structure of the application. So there's no reorganization or rewriting needed at all for this.
For the A1
Right.
application. Okay, that's helpful. And then for our NCI and NIA colleagues, I know we had a brief slide, but if you wanted to take this opportunity to, if you wanted to expand a little bit more on, what you're interested in, tools for the Pangenome, we have a few minutes now.
I'll let maybe Marilyn talk about what NIA has been doing in the field of Alzheimer's disease genomics with the ADSP that she started 12 years ago, and what kind of data we have in the repository in IGATS in terms of genetic diversity, and how we could benefit from new pangenome informatics that would help the adoption by our research community. Marilyn, maybe if you can.
Yeah, sure. We just had a big data release, and right now there's 62,000 whole genomes, and it's a globally diverse ancestry population. So we are very interested in diversity and sifting through the various components of, generally to get to why did these populations show different abilities to be protected against the disease or to be more vulnerable to the disease. We have problems in a big way with structural variants, so we sure need a lot of help there. We need population-specific variants because that's going to drive us towards our therapeutic targets. We obviously, our long-term goal is to find therapeutic targets. So if we can use the human pangenome to do that in an accelerated way, that would be a major goal obtained for us. And if this could be a launch point for that, it would really help us in an extraordinary way. We also understand things like genetic drift in our populations. The way Alzheimer's disease is expressed in various continents is completely different, even though it seems like they have the same basic underlying population substructure. So there is really a lot of information needed. And I think that this initial effort on the pangenome will get it started. I hope it leverages us to move forward more quickly with a larger footprint in the pangenome, so we can transit quickly from the Human Genome Project's HG 37, et cetera, to Pangenome.
Okay, for NCI, National Cancer Institute, we have a pretty broad-based portfolio supporting cancer research, all the way from fundamental research to improve our understanding of the mechanisms of cancer development, dissemination, and resistance to therapy, to translational, clinical, and populational studies of cancer. With that said, for this particular funding announcement, we are specifically interested in analysis methods that are targeting the somatic mutations. If an investigator is solely interested in analyzing germline mutations, that wouldn't be deemed responsive from the NCI's perspective. However, if you're interested in developing methods to analyze both somatic and germline variants, as long as you include a somatic component, that would be deemed responsive.
Great, thank you. I'll maybe turn it over to, I don't know, Ian, I saw that you had your camera came on. Was there anything else that you wanted to add from the SBIR, STTR NOFOs?
I think the only thing I'll add is just to alleviate if there's any confusion about this. So we don't have the same institute partnerships for the small business funding opportunities. So this is an NHGRI only funding opportunity, but I mean, everything else I said earlier still holds in terms of, what we're looking for and the eligibility and whatnot. So I will follow up by saying if if you're a small business or know anyone interested in this, please reach out to me, and I would love to talk more about it and how we can, make things work for you. So thanks.
Great, thank you, Ian. And we have some more links posted in the chat as well. So I'll just make just for the budget, application budgets are limited to 400K direct cost per year, and the project period is for 3 years. Oh, okay. Does the NOFO support MPI or multi-PI application? Yes. So we'll keep this open maybe for another minute. If there are any last minute questions. And you're always welcome to email the contacts in the RFA if you have any additional questions. Can all MPIs come from non-US institutions? I think so. I think, we said that foreign institutions are allowed, so yes. We'll give it till about 2:55 and then if no questions come in, then we'll call it. Okay. It's 2:55. I think we'll call it. I haven't seen any additional questions in the Q&A. So thank you to the panelists and to everyone who joined the webinar for your questions, and to our colleagues for taking time to answer your questions. Please email us if you have any further questions. And again, we will post this recording on the NHGRI website probably in 2 to 3 weeks. And that concludes our webinar. Thank you, everyone, and have a good rest of your day.